Right. So, okay. So the next speaker is Dr. Raouf Issa, who is right. Sorry. Who is a research fellow in the Professor Claudio Alonso's lab at the University of Sussex. And so the Raouf started his university studies in Togo, in Africa, so very close from Nigeria. And after finishing his farm degree, he joined Dr. Serge Berman's lab at ESPCI in Paris, France, for master's degree, and he did the PhD at University of Pierre Marie Curie, investigating the cellular and the molecular basis of aging and the neurodegeneration. So today, he is gonna talk about the undertaking neurological disorder research using Drosophila model in Biortc.
Okay, thank you, Takeshi, for the presentation. I'm going to share my screen. Okay. Is it okay? Is my screen displaying?
Yes.
Okay. Okay, it's fine. Okay, thank you. Today I'm going to talk about how we could use Drosophila in Biortc to forward the understanding of neurological disease in Africa. So, first of all, what I will know about neurological disorder. The neurological disorder is an umbrella term that encompasses disease that affects the central nerve systems and the peripheral nerve systems. And according to World Health Organizations, those diseases are the leading causes of disability and the second cause of death worldwide. Those diseases are composed of more than 400 diseases, from migraine to Alzheimer's diseases. This said, the question arises as what is the situation in Africa? In Africa, the top 15 neurological diseases that are present in developing countries located in Europe or located in America are also present in Africa, as you could see in these pictures. And the top leading disease is related to the stroke. And those diseases are a mixture of communicable disease and non-communicable disease. The communicable disease, for example, are related to disease that are due to the infections, like tetanus or like meningitis, for instance. And at the side, we have non-communicable disease, such as Alzheimer's disease and Parkinson's disease. So those diseases, in contrast to other countries that have a system to handle those diseases or to manage the pathophysiology that are associated to those diseases, in Africa, those diseases are very little. As you could see in these pictures, those diseases are creating around 200 to 30 deaths between 100,000 in the populations. This means that those countries located in Africa must define the program to reduce these burdens and also develop the research strategy to increase knowledge on these diseases. So, where is the research on neurodegeneration disorder in Africa? In the recent evidence published by a group in Nigeria, and also a very recent publication published by Mahmoud, and in collaboration with some trained members, especially Lucia Godino and Tom Padens. They report in this publication that most of research on neurological disorders are focusing on neuroepidemiology and clinical symptoms reports, or on screening of endemic medicinal plants on the wild-type animals. There is, in those studies, there is less study on transgenic animals. Why transgenic animals? For example, we know that in Alzheimer's disease, what is the genetic causes of those diseases, for instance? The same thing also in the Parkinson's diseases. To be able to understand what are the mechanism of those diseases, you need to work on transgenic animal, not on wild-type animals. So all these arise the question as how to transfer the current study done in Africa into the fundamental basic in vivo research. The way to do this is to develop the use of a suitable transgenic preclinical models. There is a lot of preclinical models, but in the Africa context, there's many things we may need to take into account. The first things is the genetically, the model we need to choose must be genetically amenable. The second things is this model must be shaped, because as we know, most countries in Africa are developing countries. So if you have to use the model that will ask for a lot of money, it will be difficult to at least to populate this model. And the second and the third point, which is not the least, is the model we need to use must be in accordance with the ethic of animal experiments, which is currently very required by many funders. Therefore, as my case, I suggest the use of Drosophila melanogaster as a model among other models that could be populated in Africa. Why Drosophila? Drosophila because Drosophila is a good system to study the biology of nervous systems. As you could see in these pictures, there is many functional area present in Drosophila that have their homology in the human. Especially there is area in Drosophila that control the learning, controlling the vision, controlling the smelling, and also the movements that are also present in the human. The second point is that there is a conserved molecular and cellular components. For instance, we have the presence of dopamine in the Drosophila, and also therefore the dopaminergic neurons, as we could see in the brain of the Drosophila here. We also have some subcellular components of the neuron, like axon, that are present in Drosophila, that are targeting the muscles. All these make a Drosophila good to study the basis of human disease, to dissect the neuronal circuit functions, and to understand the genetic network underlying those diseases, and to use Drosophila as a principle for drug screening. To materialize what I just said, I will focus on one disease, Parkinson's disease, which is a movement disorder. Parkinson's disease is characterized by the loss of dopaminergic neurons that innervate a specific area in the brain of humans, mostly area that are composed of chordates and putaments. And the loss of those neurons is due to the accumulations of cytoplasmic inclusion called Lewis bodies. The Lewis bodies is an aggregation of a protein called alpha-synucleins. And how alpha-synuclein is aggregates? The alpha-synuclein is aggregates because of the impairment of a component of intracellular pathway called autophagy. In the normal time, alpha-synuclein is incorporates inside the lysosome through a receptor called LAMP2A. But in the disease, like Parkinson's disease, there is a loss or a reduction of these functions, leading to accumulation of alpha-synuclein that are observed in patients suffering from Parkinson's diseases. In Drosophila, it's possible to model this disease and to model the accumulation of alpha-synucleins. And during my PhD, I have used, for instance, Clembin assay. In this video, as you could see, when you tap, you startle Drosophila, you will see that those flies are able to climb from the bottom to the top. And animals that are not able to climb will stay in the bottom. And this video, in the middle, are flies that are expressing alpha-synucleins. And it's possible to quantify the climbing abilities and to make a graph. And here, for us, here, I show that in association with my colleague, Remsberger, that the expression of alpha-synucleins in a specific neuronal population lead to impaired the locomotion, as you can observe in yellow here, compared to the control animals. After observing this, we dissect the flies expressing alpha-synucleins to understand what are the neuronal basis of these impairments. And we found a subpopulation, as you can see in this image, a subpopulation of 15 neurons that innervates a specific area in the brain of the Drosophila, painted here in pink, called mushroom body. What we observe about those neurons is that when you express alpha-synuclein in those neurons, this induces the loss of projections, as shown in this picture. Here, this is the projections of neurons that express alpha-synucleins, and in the top, this is the control. As you could see here, there is a reduction of those projections. The second things I have conducted during my PhD is to show that when you increase this receptor called LAMP2A that are involved in the transportation of alpha-synuclein into the lysosome, we are able to reduce the accumulation of alpha-synuclein here, as you could see in these Western blot pictures. And subsequently, we are able to reduce the impairment of locomotions as observed in these histograms. Collectively, those data enable us to propose a mechanism of alpha-synuclein leading to movement impairment in Drosophila. In the top, we have a normal fly that are able to climb into the top. And the bottom, we have a disease fly expressing alpha-synuclein that are not able to climb. And they are not able to climb because there is a presence of alpha-synuclein in sub-neuronal populations that innervate a specific area of the brains. And if you compare what we have observed in Drosophila, we could see that there is an homology with what is observed in the human here. One thing we could also do in Drosophila is to study the toxicity of environmental toxin. Environmental toxin, one of the environmental toxin is a paraquat. And the paraquat, even forbidden in some area in the continent, they are still used in many farms in Africa, like herbicide. But those toxins are those substances are very toxic. And I demonstrate that when you expose Drosophila to those paraquats, this leads to the lethality in the frame of 72 hours. At the cellular level, there is a modification of dopaminergic neurons. As you can see in this image, flies that are being exposed to paraquats have a modification of the size of dopamine neurons compared to the control one. From this, we'd also define the neuronal mechanism through which the paraquat induces its toxicity in the Drosophila. And we found that this toxicity go through the receptor of the dopamine that are present in the glutamatergic neurons here. And when we reduce the expressions of this receptor in the presence of the paraquat, we are able to rescue the lethality. We are able to rescue the lethality that are being induced by paraquat alone. From these observations, we try to mimic pharmacologically what can be the protective effects from paraquat when we expose fly to the to dopamine agonists for choline and IBMX. In the presence of paraquat, we found that this is sufficient to rescue the lethality that are being induced by paraquat alone. Because the dopamine receptor is a GPCR receptor, it increased the cytoplasmic calcium levels. And inside the cell, there is a receptor that are involved in the storage of calcium in the cell called rhinogenes. And we find that in the mutant of the rhinogenes, we found that the mutant of rhinogene are resistant against paraquat toxicity compared to the wild types. Collectively, our results enable us to propose the mechanism through which paraquat induce his toxicity. And we suggest that there is two ways. There is one way of toxicity. When you are exposed to the paraquats, you may induce a high oxidative stress in dopamine neurons. And this will increase the release of dopamines and the increased expression of the dopamine receptor in Drosophila called DAMP. And this subsequently will increase the calcium level in the postsynaptic neuron. Here, the postsynaptic neuron of dopamine neuron is a glutamatergic neuron. And this ultimately will lead to a neuronal death and therefore a death of Drosophila. But in contrast, if you suppress the expression of dopamine receptor in the surface of postsynaptic neurons, you may be able to rescue the lethality. At the side of what we could do with Drosophila in terms of the study of neurological disorder, there is other studies coming from colleagues that show that we are able to study disease like seizure and epilepsy in the Drosophila. We are also able to study the traumatic brain injury in the Drosophila. And an interesting point also is that Drosophila can be used to study the protective or the toxicity of medicinal plants that are very used in Africa, as elegantly highlighted by previously talk from Professor James Olapade. So this is also an invitation for people who are interested in Africa to study what could be the relevance of medicine plants to try to use the Drosophila. And then I think that Biortc will be the place to create that collaboration. So from Drosophila, therefore,
Raouf, could you start rounding up?
Yes, I'm like at the end of this talk, yes. So using the Drosophila, we could then study the response to environmental toxins, understand the behavior and the lifespan of Drosophila that are being exposed to that toxin or that are expressing the pathological form of genes that cause a disease, and dissect the consequence at cellular levels, and also analyze what's going wrong with tissues phenotypes. So to sum up, one thing that are now true is that there is a high prevalence of neurological disease in Africa. So by introducing Drosophila as research models in Biortc, we may be able to dissect out the pattern of the disease susceptibilities, to identify environmental clustering, to define suitable medication response at the population level and at the individual level also. And this will ultimately increase will have an impact in people studying using Drosophila as model to increase their knowledge in how this disease develop and what is the risk factor. So to finish this, I would like to at least acknowledge my formal PI in Paris, France, and current PI where I'm doing my postdocs. Thank you.